So, already the 11th lecture out of the series of 14. We're over halfway. So today, I'll go you in on sequence motifs and algorithms, sequences, greediness of methods, exhaustive methods, and things like that. First of all, apologies for being late. I was stuck in the elevator in the other side building. That was interesting. And something else. It was tight, but then it became more than tight. All right, so quite a list. What you see in red, there is a way out. You might survive this. So, bit of profiles, what the philosophy is, what they can do, what they cannot do. Then we'll go into motifs, and motifs are really important in anything to do with generical control. Gene regulation, so I'll linger a bit on that, and I'll fill you in a bit on the biology, for those who need it. Then we go firmly into motifs, what are they, what can they do, and then we go through a number of approaches to recognize motifs and to get a handle on their biological meaning, and a whole cascade of methods are in this field, and we'll cover the most important ones, but it will be highly unlikely that we'll finish all of this today. So the first half of next lecture will be on this as well. So the first hour on Friday will be on this, and then we go over to pattern recognition and cluster analysis and all that. Okay? Good. So, profiles. You've worked with profiles, knowing it or not. You've run Cyblast, that is firmly based on profile technology, and they're called their PSSMs, as you know. So, profiles, of course, represent blocks of aligned sequences. And, basically there are various ways of doing that. They can be elaborate. You've seen the blast scoring. You take the log of propensities and of ratios as compared to a random model, and so on. You can take simple frequencies and all that, so you can make it. You can use high HMN profiles, since the last lecture that's to tell you something. So basically, they can also include position-specific gap penalties, so treating the gaps that might occur in alignment in a way that you get more control over them. And but importantly, anything in sequence analysis, well, the vast majority of methods in sequence analysis conform to identically, independently distributed model of sequence alignment, meaning everything is in fact independent. So we can just multiply the probabilities of all of these events. And that has a few consequences. For example, it might well be that if you study a protein family, that you observe something like, well, if there is a leucine in position 3 in the alignment or in the family, then a tryptophan becomes highly unlikely at position 5. So if you have these sort of relationships, how can you build those in a profile? Well, the simple answer is you can't. Everything is supposed to be independent. And these things can be very important. You can think about a way of still doing it. You might think about, in Markov models you have first order, second order. What if you would take not just a single position, but describe everything as little regios, go to 3 positions or 5 positions or something like that. Then you will be able to capture something like that, but that is certainly not standard in profile technology. So basically, the answer is no, you cannot. And that might be a problem. You do not capture that kind of information. Another thing is, with all of in BLAST, you have that if profiles or if sequences or sequence alignments become short, covering not too many amino acids, residues in general, then your statistics start breaking down. It becomes difficult to get any good signal. And short alignments should just score 100%, and then yet you still are not sure that you have a pattern at hand that is any significant. So statistics will not help you anymore. And another thing, of course, is that you have reduced signals, a low complexity that can also severely reduce the profile sensitivity, certainly if most of your sequence happens to be low complex. Still, this technology is really used a lot. We have the PFAM database, and there are many like that, big resources, important ones that are, in an important part, really based on profile technology. But, you know, to the rescue perhaps, or to help, or to highlight advantages of profiles, they can, in addition to sequence information, also capture sequence-derived information, such as secondary structure or structural features. To, you know, you can upweight things and do things. So it's not that you're totally without any power using profiles. And of course, you know, the latest is hidden Markov based profiles, such as they are in the PFAM database. Another point is, if you compare a sequence against the profile, so you add a sequence to a big alignment by comparing it to the profile of that alignment, and then if you look at most technology, how symmetrical is that? For example, the chance that you get a gap in your single sequence as opposed to a gap in the whole profile. And there you often find, it's not symmetrical. So the technology that has an influence, and that is something you should be, you should realize that that can happen. Yeah? So a lot of technology is asymmetrical. That might be important. So then, you know, if you would like to do something else, then you can resort to, more generally, to pattern recognition techniques, and here in this case, pattern matching, to capture the information in a profile. Maybe you want to zoom into a more local situation, or you would like to capture this situation. If there is an L at 3, there cannot be, or there should be a P at position 10, or whatever. Those things you might, if they're important, then you might try and find technology that can capture them. And so pattern matching is generally used, and this lecture will be part about that. And certainly the regular expressions that are part of this. And here a small example. So proteases, these are little nasty proteins. In fact, you know, these are proteins that chop up other proteins. It's important. We make use of this after every meal. But, you know, since they represent a sense of danger in your stomach, for example, these proteins are locked until they're really needed. And then they're sort of released in their functional form. And these proteins, the hallmark of these proteins is so-called catalytic triad. Their active site, the site at the protein surface where its function happens, is comprises 3 amino acids: histidine, asparagine, and serine. And you know, so they're spread over a sequence. 3 amino acids that are very important. How do you think you would capture those with a profile? You know, it's not enough signal. So if you want to find that, then it's a good idea to really use technology that really zooms in on these amino acids, for example, and maybe the spacing in between or something like that. Right? Okay, so one of the examples today: transcription factors, proteins that are important in the transcription control. They are basically, you know, they switch on or off a polymerase, the copying molecule that converts information on the DNA into a messenger RNA sequence when it comes to protein coding. And they do many other things also in non-protein coding processes. So what they all do, you know, involved in all this is the binding of those molecules, interaction, very important, between various transcription factors, but also, of course, between the transcription factor and the polymerase molecule. Initiation, enhancing, and repression are all things that play a role to control the switching mechanism. These transcription factors come in various guises. There are, you know, 40, 50 different folds known. And basically, the idea is that, for example, in our body, there is a single copy molecule, and that's of the type polymerase 2 in our body. So basically, that is the thing that copies DNA into a messenger RNA sequence. But the switching on and off is by means of transcription factors, so you have very many different switches. Proteins that come into play for a specific family, gene family, and maybe another transcription factor will play a role in another switching mechanism. And so that's why it's interesting to zoom into these, and finding where transcription factors bind. That's a short, localized motif expressed by DNA is very important to know, and that's why a lot of technology tries to establish that. Okay, yeah, and of course, you know, necessarily a transcription factor consists of a DNA binding domain. It should bind, of course, to the DNA, and also should interact with the polymerase. And that is done by means of an interactive domain. Here you see that a bit more schematically, the picture. And you've seen this before, I think, in early lectures, so we can be quick on this. So here's a transcription factor binding site. the transcription factor is supposed to bind to that binding site and at the same time interact with this polymerase type 2 molecule, which should in itself bind to a motif that is TATA on DNA, and then the transcription process might start. And then you see another thing, that DNA is sort of protected by nucleosomes, chromatin structure, and that has also local rules. And then it can be that a perfect transcription finding binding motif is sort of locked in place and is not accessible to these molecules to bind, and therefore they cannot have an influence on the switch at that moment. This one might have it and so on. But all of this influences the regulation process, because what also should happen, you can see that here, that the DNA should bind in order to facilitate the binding between this transcription factor that's bound here and the polymerase. And if you have all this stuff in between, it might be that the curvature won't work. And all kinds of physical things that have an influence. And all this is called epigenomics. Right? It's not the DNA per se, but the things that happen around DNA that play a role. But of course, the information contained, why does this thing bind here? It's clear, of course, that the composition, how DNA is formed, what their base construction is, still has a big influence on all that. Okay, so it's clear that this is just summing up, they're important, and this is all of what I told you. Okay, this is just from the book Essential Bioinformatics, a little picture to show you the difference. Basically in bacteria, you have one transcription factor, one protein that is important for this. But in eukaryotes, cells like ours, you can have a whole complex of possibly many transcription factors and related proteins that together provide the switching mechanism. And here is the geek view. For a computer scientist, you want to see the world like this, right? So here you go. These are all the things that happen in the process. Okay. You have a gene and a protein, and there is expression, and then the gene is linked to a certain site. And this is all stuff that really is functional, that should happen at the same time. Good. So, what technology do we have to really measure? Because, we're all now, calculating stuff, but we should base all of that on knowledge. And luckily, technology has helped here as well. There are two main techniques that are important in this business, and that is chip-sec and chip-on-chip. And so basically, this gives information on where, so this is the real thing, this is a transcription factor, GAL4, we'll come back later on I think. MIG1 is another one. They appear to bind on motifs that are here. You see that the DNA structure is also important, because that the three-dimensional structure, of course, this is on one side that can be bound. If it's on both sides, then the molecule should sort of be able to curve around it. So basically, these motifs that bind to GAL4 have to have sort of spacing that is defined, and some really conserved parts of it. So this is the binding site in the GAL4. chip-sec is a technology that is able to really establish where on the DNA such a molecule might bind. And that works like this. This is, I think, a picture from a Nature from a Nature paper. I think it's Nature Technology or something like that. The link should be somewhere. Oh, here it's Nature Reviews. Okay, fine. So you have an so what is the trick? You get DNA where a transcription factor is bound. That's of course important. Then the protein you put the protein in. It binds to the DNA at the proper position, hopefully. Then you establish. Then you find out where that is. You cut the DNA around that protein binding site. So you have the bit, the DNA, the protein binds the DNA. You cut it around, flanking in the flanking region. Then you take that stuff out. Basically, this is all sort of elaborate molecular technology, of course. I'm just glossing over all this as an innocent bioinformatician. So you take that out. That's easy, sort of fishermen do. It's a bit like that, but not really. It's a bit more cumbersome than that. So, but then you take the protein off the DNA sequence, and now you have that small DNA fragment, and then you sequence that fragment, and then you know at least how really that fragment that binds the protein lies, where it is. Do you know the exact residues where the protein binds? No. You have to do more, but this is at least you can identify really where on the DNA does this protein bind. So there are a few problems with that. And that is, so here you see what sort of information you get out. You can read this as the region of the figure. So basically you get information chip on chip, and chip sex is a bit, chip sex is this technology you see here. If the peak is high, there's a high probability or a high binding score of that protein that transcription factor binds in. And there are various controls are possible what you would normally for this protein expect over DNA, that's then this, and then you can say, this binding peak is really significant, so it's probably a real thing, right? What is the basic problem with that? Yeah, here you go. These binding sites are very small. So, how likely is it that you have just a GAL4 motif, 3 conserved letters, spacing of, say, 7 to 15 in between, and then 3 conserved letters? How often do you think, over a sequence of 3.3 billion letters, such as we have, that would occur just randomly? Many times. So, that technology, would you say, that binds at the same. So what is really biologically important and what not is a difficult thing. This technology doesn't say anything. But then, so that's the main question. Has anybody so far an idea what could you use? Humans are not the only species on the planet, right? So what would be an idea to do? I'll throw a suggestion in. Suppose we have organisms that are a bit like us.
Conservation.
That's a magic word again. If you are in this course, you need to say an important word. There are 2 possibilities. If it's not homology, then it's conservation. And, yes, very good. So, absolutely. So if it's conserved, so if you find spots where that binding site is more conserved than somewhere else, then you have maybe a clue. Is it then 100% sure? No, but at least you have something in hand to discern and to get a better handle. Okay, so basically, transcription factors are important to establish a relationship between a gene A and some gene B if the same binding site would be close to this gene and maybe related to this gene, and when the same holds for that, then you can say, hey, that means that the same transcription factor will bind, so if that thing is available, both of these genes might become expressed, and why would that be? Maybe because they have to cooperate, right? It's all guesswork, reverse engineering, Sherlock Holmes. But okay, if we have no better evidence, we need a Sherlock Holmes. So that's what it mainly is. Okay, and for drugs discovery, for example, this is really very important. That's why big pharma is all doing a lot of research in all this to find these relationships to make use of that and produce drugs. Okay, so basically, how can we abstract these binding sites into a gene regulatory network? And so here we have this standard book picture. This is this a prokaryotic or an eukaryotic gene, by the way? You're not sure. It's Monday, I know, start of the week, but you know it's eukaryotic, right? Because you have introns and exons. Promise me that at least. Okay, that's good. So then here, the next picture, same thing. Now we have regulatory elements. Think for a moment, transcription factor binding sites, and you see that all kinds of proteins are in competition to bind here, and but together they establish something that regulates the making of that gene. And see what can even happen here. In the gene, you might have molecules that have a tendency to bind there. What do you think would that do to the polymerase that tries to slide over the sequence in order to faithfully copy the gene region into a messenger RNA? This very often is inhibitory, right? Okay, so here you go. This is then the process. The red blob is this polymerase molecule, the central copying device, on its way to produce the messenger RNA molecule. And so here you already see something. If you look carefully, you see here are arrows, and here you see this thing, the end of the train track, right? What happens with the train, it bumps into it, so it stops, hopefully at least. And that is also the idea here. So this signifies inhibition, and this, of course, signifies activation. So if we take this, and these regions, by the way, incidentally, are called proximal or distal, depending on where they are, before, during, or following, after a gene, and they're bound by all kinds of complexes. And if this would be the situation, this is, by the way, this gene here is a transcription factor itself, because transcription factors are proteins. they should be encoded by genes as well. So you have transcription factors that are important for the formation of transcription factors, of course. They're interesting for gene regulation. So if you have this, that means that this transcription factor, if it's produced, has a negative influence on this gene and might have a positive influence on gene C here. You see that? So then the thing should, of course, be produced itself in order to become functional. So if you have this, then you can abstract away from this into this gene regulatory network. And again, I already said it, I think, many times, this network you don't observe in cells, right? It's an abstraction for us to think about it. So this is basically the biological reason that you can abstract away from this and generate this network. Yeah? And you can see that, if you check it, that this molecule, and I think I've shown it before, so I can go on. Just a bit more fun. These transcription factors look beautiful very often. That's a nice thing to know. I'll show a few pictures just to hopefully please you. And so, we have 3 kinds, helix-loop-helix motifs. One of my favorites, the zinc finger, and then another one is a leucine zipper. And you see, this is two helices basically attached to one another, based by the interaction of leucine residues that therefore have a regular spacing along each of the helices. And this here, you see, this is a small mini protein in a way, where zinc as a metal is important, and then there is a requirement in these sequences of these pertaining to these folding into these structures that you have 2 histidine residues here and 2 cysteine residues there. These are the pictures, but I'll show you a bit more. This is a helix-helix group, so this stuff here is DNA. This is a protein, well, a schematic representation. Hopefully you can appreciate that this protein is pretty much alpha helical protein. More in the next course, but here you get a preamble. And then there is another one here. This is the zinc finger, and it's really a finger that pokes into the DNA. It goes into the major groove. And then, you have 10 fingers, but most molecules here have 9 fingers, and all these 9 fingers sort of articulate the DNA. And how that works together, is sort of binding. So you can very well regulate the exact binding mode, how strongly you bind. Because there are also zinc fingers that have 13 fingers, so it's far more versatile than we have. Question to you.
The grey dots are the zinc?
Sorry, that's you said, but the answer is yes, zinc. That's the zinc. Yeah. So each small mini protein here and here have a zinc atom. This is needed because small proteins, I think I've said this already, but I'll do it again. Proteins are normally, hydrophobic or philic on the outside. What was that again? Philic. Yeah, they love water, right? Hydrophilic means you love water. So you do that on the outside if you're close to water. It's always said when you love somebody who is not available, right? So it's better to love it when it's there. That's why stuff on the inside of proteins, are hydrophobic. They don't like water and want to get away. That's more logical to do. But small proteins really don't have so much an inside and an outside. It's flexible, so water can nearly get everywhere. And that is why they tend to be very unstable. So to reach a minimal degree of stability, very often they make cysteine bonds, for example, is one of the things you can do, or coordination with a metal atom. And here's one very nice example. And if you look at this, you saw that on the last, these residues are important, and this is roughly the spacing you'll have in such a sequence. Now, suppose you have an alignment of many zinc fingers in different organisms, and then I can tell you all these residues look like nothing. Totally, mutated away. What do you think of your alignment? Very difficult. And but there should really be CC. That's normally there. But the alignment methodology is not upweighing this enough to bring that out that you get an anchoring of these positions. So you need maybe some other technology or a very good multiple alignment program. I won't say anything. But there is one where there is some tools. It's called Praveen, by the way, where you might be surprised to hear, where there is some functionality that you can use to do this. All right. Anyway, zinc finger. This is all there is to learn about it. Here are 9 zinc fingers, in 11 there are 13 zinc finger molecules. Okay, then the other one. Oh, this is the story. You can read it at home. Here. This is a nice one too, right? It's like scissors. It doesn't cut the DNA. That's not the idea. It binds it. These are these 2 long helices that can sort of move out and bind the DNA at either side of the helix. The DNA goes into the plane, right? And So this is the leucine residues. You can't see them are roughly here. And why is it regularly spaced? one turn in helix, how much was that exactly? Who knows?
3.
Better, come, you can do better. 3 point what? 6. You should know this before. 3.6! That's fantastic. Do you know full circle? How many degrees? You know that? 360. Hey! Any idea? So what angle is there between two residues in a helix, you think? Guess, if you have 3.6 divided over 360 degrees, you've got a nice answer, right? Very nice round number, I think. But that means then that the spacing, if it's 3.6 residues, that 2 turns are roughly, well, 3.6 times 2, let's say 7, right? And that is the spacing you normally see. Start counting. Hopefully I'm not talking out of somewhere else than math here. So this is roughly the spacing. It's therefore fairly regular, because the interaction between the two single residues has to be every other time. Yeah? Nice molecule. So you see the beauty of the protein world. So I'm now talking you into wanting to attend the next course, right? In February, that is. The sequence analysis course. In January, there's no saying no to you. But February, you could say, I jump over to systemology, you could get away from that course. But that's why I'm trying to advertise it a bit, right? Okay, so another overview picture of all of this, what happens where. This is the nucleus in the cells, in eukaryotic cells. In bacteria there is no nucleus. Basically everything is just spread over the cell. But here there is an organization in a eukaryotic cell, a nucleus, where the transcription basically happens. A messenger RNA being produced in the nucleus, get through the nuclear pore in the outside world, on its way to a ribosome, to be able to be translated. So translated happens by means of ribosomes, cell organelles. Okay? And then after this a lot can happen. It binds to something that switches off immediately, or there is a molecule, a compound that is attached to the protein, helps it functioning better. All kinds of things are possible. And you know microRNA? Have you heard about that? It's a little nasty molecule in a sense. Because what is it doing? You've had all this trouble, happening here, and the messenger RNA is produced. It's on its way to the ribosome in order here to be translated into the protein. And then a small RNA molecule comes that sort of binds locally to the messenger RNA. And this molecule now has a bump on it. It goes to the ribosome. Sometimes it's recognized by the cell, oh, there's something funny going, it's taken out. Or it won't work on the ribosome, because that stops really the physical process by, again, molecules that sense the messenger RNA and produce another sequence, namely a protein sequence. Okay? All right, so all right, maybe a fair enough now to tell that many processes are really, have something to do with local sequence motifs there, where sequence analysis is not the forte to get information above the table. So we're going, this lecture is mostly on motifs and zooming in into smaller segments. Okay, so and we do most of this based on DNA, because fewer letters, easier to explain, and a lot of work is happening at the DNA, RNA level. So what are nucleotide motifs? So motifs that take place on DNA or RNA. They normally, are very short, 5 to 16 letters, so not very long, maybe 20. And microRNA itself, this other nasty molecule, is roughly 22 in plants mostly, but say up to 25 letters. So it's all short stuff, right? But they still may contain gaps if you compare them, obviously. So there is spacing possible or necessary sometimes in order to really compare them. So what are examples of these motifs that have functional importance? Splice site recognition, I'm treating it. Start-stop codons, obviously. Oh, and in the middle of centromeres. Is that all known by everybody? Okay. DNA molecules attach and to one another, and they do this basically by one sort of central point, roughly, well, in the sort of closer to the middle of a DNA sequence, not quite, but okay, and there they attach. And it turns out that around centromeres special stuff is happening. Basically, I don't know if I told you, but during the Human Genome Project, the battle, Greg Venter, Darth Vader, told you the story, I think. Basically, at the first version they had only 70% of the DNA sequences. The rest they couldn't get. It didn't want. It was not amenable to sequencing. They didn't want it. That was around. The centromers were really difficult to capture. So what do you say if you're a real scientist? You have technology developed. You're measuring and you know, oops, these regions, these sort of regions, I won't be able to say anything about. What are you going to say? You're not only a scientist, you're also human. That's so important. Obviously. Why? Because I can't measure it. I see. Okay, very good. And then, of course, a few years later you find out and they can, holy hell. This is where all the important stuff starts. This is where beauty lies and AIDS in people, we should have zoomed in earlier, right? But okay, that's the happening. Okay, and the protein motifs, obviously, transmembrane. These are the membranes, the bits of a protein that go into a membrane, the outside of a cell. They anchor there, so that sort of mechanism is also very important. Membrane proteins are very important for drugs and medicine because they transport what happens in between cells, and that is normally important. Well, smaller sites and proteins, phosphorylation, what compounds can bind to a protein that has certain requirements. I think glycosylations, glycosylation sites I've shown you in an earlier lecture. Otherwise I will again. And so there are various functional sites, active sites, binding sites, all kinds of stuff. And of course, you saw already one example of this. The leucine zipper is an example of so-called coiled domain. Right. So this is, if you have you can have a single site that is a certain family or has some functionality attached to it. You might have a set of sequences that are related doing the same thing. Maybe a single transcription factor might bind to all these variations. And then what you try and do, the same as before, an easy way to represent such a group is by a consensus sequence. And then you might say, okay, the best consensus here, if you look at it, might be this sequence representing all of these here. Or you do something funny, you see that here? What's that letter? Have you seen that earlier on? No? Absolutely. So here you are. If you have 4 bases, these are things that are, this is what people have established. If you have an R, that means, according to these standards, an A or a G. So this is a redundant, a reduced alphabet you get. This. R is A, G, or G. And Y is C or T, and so on. Yeah? And in protein sequences, you have also a few of those letters. So, and if you're here, if you're 4 folds, you generate, if you know nothing, that's an N. And I think I've shown you a DNA sequence once upon a time, asking you how good are your eyes. And then you saw N's in there, right? So there was this N. It's an N that represents, there is a base, but you don't know the identity of that base. In proteins, you have the same sort of thing, where you have, well, an X is for, there is an amino acid, but the technology didn't allow me to judge which one it was. And there are 2 amino acids that are, certainly when you do crystallography, the older technique, hard to discern, and that is an aspartate or an aspartic acid, and the same for glutamate, glutamic acid, and so therefore they created the letter B and Z to establish that fact. So if you see those in the sequence, it's either it's D or E, and Z means N or Q. Okay? Okay, so now we go a little bit into something, and you've used all of this, maybe without, if you've ever used Unix or Linux, I hope you have at this point, then you know you're using all of this stuff. Have you ever typed, what is it, RM star dot star? Basically you shouldn't. But then you're executing a regular expression, and in a moment you will see what that might mean, right? I had once this advice when I was really totally innocent earlier on in my career, when I had a little project, I was helping guys, and that was some regrettable consequences. No backup, and so on. Week of work gone, programming like crazy, and then some, I won't say the word, advise you something like that. Okay, anyway, these are the letters, and you have strings, and these are symbols that are composed of the letters of this alphabet, and generally in DNA we have a 4-letter alphabet, well, disregarding these reduced alphabet letters. And a regular expression, you can also write like this. Regex for now means regular expression, is a formal method to define subsets of strings or to define some kind of formulation of a string where a defined subset of possible strings adhere to. And here you have an example, and we'll go through a few of those things on the next few slides. These are used really very widely for pattern matching, and also in sequence analysis they come in very handily indeed. Yeah? And what you can do is, if you have a regular expression, you can just say, "Ooh, if this represents some important motif, I have a protein sequence. Is this regular expression part of the protein?" Right? So, could this protein have this functionality? These type of questions you can then ask if you have these regular expressions. So, this is what they can answer. I want to see all sequences that contain a C. That's an easy regular expression to test, right? Is there a C? Or contain a C or an F. Or contain a C and an F you can ask. So all of these questions you can ask by using a regular expression to fill in, to define that exact question. And this one can also be done a bit more complex, but you can do this with regular expressions. So, what have we got? We have symbols from an alphabet, right? So this is the collection with the letter C. It's a bit set theory. And if you do an operation on the regular expression, you get basically a new regular expression. It might be filled in completely, or it might still have a few variables. And these are the functionality you can do. You can put one regular expression after another, look to the cross section, see if they're repeated, and so on. You can do all of those kind of things. So here are a few examples. If your regular expression is AC, what do we get out of this? If this is your regular expression, you're looking for a sequence that contains AC. So we get out, of course, all the sequences that have an AC as two letters following one another. And the same, of course, if you say AC, if that is your question, then you get all sequences that contain AAC as an answer. Now here it becomes a little bit more interesting. This means OR. So this means, is there a sequence that contains either an A or a C or a G or any combination of these? Okay? So you get all of those sequences back obviously. This is, how do you evaluate this? This is an A or a C followed by a G. So what are the two possibilities you can get if you ask for this? AG or CG. Yeah? So far so good. Nice. So here you get the union of things. If you have this, A or CC, well, you get A or CC in the sequences. If you get this, what do you get? AC or AC, you get these type of sequences. Here you go to repeats. This is, a plus means at least one of them, and then any number repeated. So that would evaluate like all these sequences come out of this C, CC, CCC, CC, and so on and so forth. And this would lead to these type of sequences. Any type A here, this A or C repeated many, potentially many times. So you can have all of these sequences coming out of this single regular expression. Then you can have do some other operations. This basically means in this grammar, 0 or more C's. So what can you have? The empty string, which is signified by this letter epsilon. This means empty, or a string of C. And this therefore means, if it's the question like empty or AC, then you have this type of letter. So A or is there a C with a question mark? If there are no brackets, the symbol just is for the symbol before it, right? So then you have this as an answer. So there is either no C, then you have AG, or there is one C, and then you have ACG. Yeah? And this is wildcard here. We're going to this RM star dot star now, right? And so here you see what it means. It just means absolutely, everything that has anything to do with C, but also just an empty string will do, right? And here you have the same thing if it's to be followed by a C. Okay? And here, no. Okay, so you probably get the hang of this. But you can also be more specific. You can say, I want to see 4 C's in a row. Yeah? And 2 to 4, I want to see, is it 3 minus? From 3 or further. This is not a minus, this is 3 and further up, 3 to n, and then so on. So all kinds, and this depends also on the exact grammar you use. And I need to tell you something that might be annoying to you. Different communities use different grammars to specify. So a question mark in one grammar might not mean the exact same thing as a question mark in another grammar. So you have to be aware of what grammar you're using, basically. Okay, so any other things you might do. So a dot, it means just a symbol, any symbol. So it's something else than an asterisk. So the dot asterisk, you can combine those if it's necessary. The asterisk alone would have done that. What you can have is this symbol in here. I think it means to start. No, this is not a symbol here. Sometimes I go wrong. Many grammars have that as the start position, and here it's used as not. Right? So this means not an A. So what letter have you left if you can't have an A? The 3 other ones, clearly. And here is if you not A or C, means G or T. And different groupings, and you can all combine that stuff. You can have a whole regular expression and then with a question mark behind it and so on. So that would lead to another regular expression. Okay, so here you see that thing that sometimes this signal, the little head, matches the start of a string. And this is more general most of the time, and you'll have an example later on in this lecture as well, if the dollar sign matches the end of a sequence. It's the end symbol. So a symbol that's not really there, but it signifies the end of a sequence. Okay, so how would you specify, I want to see 6 C's followed by 6 T's. Is that difficult? Well, here you have the regular expression. That's the one, right? You get it. This is also possible, any number of C's followed by any number of T's. But what can you not do? What does this mean? The same is difficult. Equal number of T's. How would that work? This n is not necessarily the same n. That's the problem. So Cn means any sort of number. This you can do. So this is n is 4. Then you have 4 C's and 4 n's, like here. But what you need to do to answer all of these, you need to expand on each of those and get these sequences. So you need to say this is then Ct and then C to the power C2, T2, C3, T3, and it becomes a bit cumbersome to express that after a certain time. So there is no mechanism to fill this into the regular expression itself. You have to expand on the regular expressions like this. Yeah? So they're not for everything, unfortunately. But they can do a lot of things and enough to be interesting for sequence analysis. Okay? If you want to go to this, you need to, this is all linguistics and grammars and Chomsky normal form. Chomsky, the philosopher, who you might know from all your YouTube films, where he predicts hell to the world, for good reasons. This is a very intelligent man. Listen to him, but it's a bit bleak, his prospects. But anyway, it's really interesting to listen to him. He has a couple of Nobel Prizes, and so a very interesting character indeed. He has also been important in linguistics. Okay, so, what can you do if you have a regular expression? Well, of course, you can find out the pattern described by the regular expression. You can learn from it. You can, of course, check if a sequence contains a regular expression. That's already what I told you. And what is very nice, certainly for computers, is that, evaluating if a regular expression is part of a sequence is something that can be done very, very quickly. So that's easy, because it's just, relating to the length of the regular expression, of the sequence. It's linear in the sequence length, so that is very good. And you've seen a mechanism, there's a program that I talked to you about called BLAST that uses this stuff. You've seen this picture before, I think, for this regular expression. So automaton theory, and you've seen this slide, I think. You've maybe seen this one, and you've seen one more important, you've seen this one. BLAST uses really regular expressions for this sort of technology. This is all regular expressions. So Unix uses it in their interface to users, and BLAST uses it in the method, and many bioinformatics methods have some use of regular expressions. Okay, here you go. What did I just say? Here are a few languages that you might have heard about. It's not only Python that is on this world, I should tell you. There are other programming languages as well. Are you surprised? Suddenly, are a few ones. And these are just commands, if you like, in Unix. Awk is a Unix thingy. Yeah? This I already told you. Do you know, in fact, the syntaxes can be different. And here is the first one of a different syntax in the ProSci database, fantastic database, having functional areas. I see some people, yeah, you're all absolutely right. So we have coffee after the slide. And apologies, talking too much again. So if you have an alignment here, check it. Maybe, and that is a task, and that is, something, is it art? You have to take a decision. What will I describe using a regular expression? So what information? And you might, for example, say, well, this sequence has been sequenced, but I'm not so sure about it. There is an S here, but I don't like to see that S so much. So you know what? If I look at this alignment, I will say, I want to see an A. You could say, well, this regular expression, they say, all you do is A or S. Look at the second position. Fair enough. But you could say, well, I'm not too sure. It's only 4 sequences. Yes, it's conserved, but I don't want to bet all my money on it. So you know what? I'll put in here just a few related to the message. You could do anything you like, right? So this is decision taking. How to convert, for example, here, you might guess what this means. This means 4 positions where I don't care. Any letter will be fine. Where is that? It's after the G. Where is that G? Probably this G. So what do you see in those 4 positions? They're all totally unconserved, all different, right? So maybe, yeah. But you still could say, well, no, this is hard to say something else if they're all different. So maybe that's. And here, for some reason, after those 4, where are we now? And you don't want, so here was the G, 1, 2, 3, 4, in this position. Basically from this user extracted that, he or she doesn't want to see a P or a G at that position. So basically, the more sequences you have, of course, the better able you are to build a regular expression that serves its need, its purpose, but there is a degree of uncertainty. But, of course, if you have a regular expression like this, then you slide it over sequences, then you've taken a decision, and based on this expression, you will find protein sequence having this abstracted motif. Here's an example. If this is over some version a while ago, I must say, of Swissprot, I think, the protein sequence database. Coffee. Sorry, I started to go after I said there was coffee. You can start. The camera is on, so only decent noises should be made from this point on. Be careful. This is all for eternity now. Okay. Any surprises at this slide? So if you have a regular motif like this, you just ask for a specific subsequence called, let's say, David. Apparently there are 71 hits, 71 sequences. Have this on board. What are you doing if you do this? Well, you get more or less possibilities. Of course, you make it less exact, so you expect more hits. It happens. And then if you push this one up as well, make that one more fuzzy, yet again you get more hits. If you do that once more, you get more hits. Once more, you get more hits. And why is this now more hits than this? Obviously because you don't demand that it should be followed, well, this E should be followed by D like there. So you expect, a better test would have been if this was day V instead of day V, right? With an I here. But anyway, you see the hang of the thing. The more specific you are, the less results you're going to get. Okay, so this, as I said, is the grammar from ProSci. Well, one of the things, where was it again here? This means in ProSci not P or G, and what was this in this earlier grammar? This little head sign, if you remember well, right? So, annoying. This is life. Okay, so there is functionality in the ProSci database where you can search in 2 ways. You can look for proteins containing patterns specified, in more than 1,000 protein families. And also you can scan the protein, the database itself, or you give it your sequence and ask for a ProSci motif. So you can either specify a regular expression and ask ProSci what of your sequences has it, or you give it a sequence, you ask ProSite all the motifs you have, what is my sequence? Okay? And yeah, here is the one that I started alluding to, but is now coming up. So this is a simple regular expression, signifies glycosylation site. So apparently you need an N and then not a proline, and then an S or a T, and then not a protein residue. That's what this means. So if you have that, chances are that will be a binding site, but if you have just a protein sequence, what might happen with this perfect binding site? Where could it be localized? If you just have a sequence, you don't have the structure. What's a requirement, you think, for a binding motif like this? Let me tell you, that it is at the outside. So if you hit a motif like this that's perfectly on the inside of a protein, it's not going to be a binding site, unless the protein unfolds when the thing is in its vicinity, or something like that, okay? Right, and here is just an example, not to read all the symbols and trying to understand them, but of a so-called extended profile. This was a paper in '96, I think. Yeah, Philip Bücher, great guy, Switzerland, Lausanne he is. He's a very modest scientist, doing much better things than he stands on stage. He's very modest. It has been very important for clarifying a very old language called Linear B, older than the Roman. You have Linear A, that's done. B is one of the big enigmas to the world. He has been important for this. He's an expert on that as well. So he likes sequence analysis, let's put it like that. Not only biosequences, but even other sequences, human culture sequences as well. Anyway, they had all done these, and then it turned out 2 years later when somebody looked at this again, this is just a hidden Markov model. So they had reinvented hidden Markov models. Would have been easier for them if they had looked at the theory of it, but they did it all from scratch, which is nice too, as a check. That hidden Markov models make sense. Anyway, another smart way to find motif incarnation, so repeating motifs, incarnation, is something happening again, and that's a suffix tree. That is a structure, you've seen it in the lecture on what was it? BWA, the method where I already showed you suffix tree in order to align short fragment to, in a fast way, to a complete DNA sequence. And so the definition of a suffix is something that's following. Here you go. You've seen all of this. And what is important, the suffix tree holds all strings that are unique in the sequence. That's important to realize. And now the question is, how would you produce such a tree? If you have a long string of DNA, how do you generate the suffix tree of that? And I'll show you a simple way of doing it. Would that be a problem if that takes an hour? Like for CWA, if you have a genome, maybe, but the thing is, if you've built a structure once, you can make use of that during many, many searches. So even if you have a bit of work to do, it's only one time, so that's not very critical. Okay, so what can you do if you have suffix trees? So you can, of course, locate a substring in S. That's what we have seen in BWA. Very quickly find the position. Where is this bit? Oh, it's there in the DNA sequence. That is a use that you've already seen happening. You can also say, and that is also BWA. What if, it's not that exact string, but a few variations may occur. That can happen in this technology. And, you could also use suffix trees to evaluate regular expressions. And that's also interesting. So where does this expression land? And that makes it, that trying regular expressions where they would occur in a sequence can be done very, very fast. Okay, I don't know if I think I mentioned that, the longest and the shortest common substring problem. What are you doing if you have all these, now we're in sequencing again, what's the idea? We have all these snippets of DNA where we have to establish the order by having overlap. And then, a solution might be a very long sequence. You repeat everything 10,000 times, that makes a long sequence, or the shortest possible sequence. So, in sequencing, they're on about the shortest common substring problem, but then again, in biology that's a bit dangerous. Why? Because we know there are very many repeats in a DNA sequence, so the shortest substring is not always the right answer, but the other side of that medal is the longest common substring problem, and historically the suffix trees have been the first feasible way of doing that. And here you see, that building trees can take a lot of storage and some time to build them. But then, if you have it, you can really use it to much effect. Okay, so basically, and I'll give you an example in a moment, what could you do? DNA has 4 letters, right? We add 1 letter to the pack, the dollar sign. Remember what that was? The end of the sequence. Okay, so that letter happens only once in the DNA, while the other of the 4, of course, happen many times. So you start with maybe the 4 letters if they occur in the DNA sequence, and then you worry after that's your top level, and then you worry about the next symbols that might occur. Let's take an example, and you just continue that process. So here we go. Suppose we have this sequence. We want to build a suffix tree for this sequence. You see the dollar sign only occurring once, and the other letters are all there in one or greater abundancy. If you look quickly, you all have good eyes, right? You see that, this is interesting, perhaps the letter C. Do you see ATC happening and CGA? Anyway, let's build the tree. We start with the base letters, that is A, C, G, and T in order, and then the dollar sign is the last of the sequence. Now, if we have had an A, take the letter A. What letters do you see following that letter A in the sequence? We'll start with the first one, a T. Any other letter? A G. Anything else? The dollar sign. So what letters again? The G, or the T, or a dollar sign. Which of these are now unique? Is AT unique at this level? Can you? No. Aha, do you see? Is AG unique? Where does it start? I hope so, otherwise my slide. AG is unique, right? Yeah. Where does it occur? What position does that start? AG, position 6. Here you go. AG happens at position 6 because that is a unique suffix. Okay? Now, the same story, of course. Check whether the $8 is unique. Obviously it's unique because the dollar sign only happens once. So there you go. That's done. And where is the before last letter? Apparently that's letter number 15. Okay? In the sequence. Sequence position 15. Now, what about AT? You told me it's not unique. So what letters will follow AT? The letter C here. There's another AT. Yes, there is. What is following it? And A. Is there anything else? No. So these are these letters, AT. Which one is unique? Apparently, ATA is unique. And apparently, ATC is not unique yet. Correct? Correct. So you go on to the next one. Right, so. So, what are we going to do in finding motifs? So basically, maybe we have a motif, then we would like to find, okay, where does it happen in the sequence? Or in what sequence does it happen? And how many times in the sequence and where in the sequence? These are very valid questions. Another question might be, I have a family here, DNA, we know transcription factors bind. What kind of motifs are shared by these sequences? So you don't have a search image, you don't know what, but your question is there anything here in this sequence that happens to be conserved of a certain identity? That's another question. That's much harder to answer, obviously. Because then you have to think about, maybe I should just generate all possible motifs. Then I say immediately, good luck, right? And check every one of them. How often do that? You can then ask that question again, where do these motifs land? So, if you're on about what kind of motifs are somewhere conserved in these sequences, maybe they should occur in each sequence. Maybe you say, well, if it doesn't happen in one of the sequences, that's fine. Or in two, then it becomes again different. You can ask all those questions. So you have a combinatorial approach and the probabilistic approach, both of which I will do. And I think, given the time, that I will not get to probabilistic, so that will happen in the next lecture. So, combinatorial motif finding methods. Very often they are sort of pretty exhaustive, so you will need a computer to do it. Here, now we go a little bit more formal, little bit more mathematical, you might say, but I'll talk you through it. You should be able to survive this. So, we have a collection of sequences. The database of sequences you want to search through is a set of sequences named S1 till Sn. You have just n sequences in your database. Right? Then there is a motif of a certain width, certain number of letters comprising that motif. So the motif runs from 1 until W for width, I think. And then some letters might be asterisk or even blank. It doesn't matter, it's a motif of that length. So we're working here with fixed motif size. Okay? And if you want to find with this method all possible motifs of any length, we will need to exhaustively go through each of those lengths. And then the question that you would normally ask is, okay, I would like, is this motif somehow conserved, or is it comprised in the sequences? And then you could say, for example, you could say, I want this motif to occur in each of the sequences, and I'm okay if there are a couple of differences. As long as the difference is smaller than k letters, I'm fine. I say that sequence conforms to the motif. You could also say, well, over all of the sequences, I would like to find something. And having distance is a distance criterion, of course, and it's just basically the number of letters. You have one string, and there's another string. How many letters are different? Or to put it another way, how many letters in the one sequence would I need to change in order to get the other sequence? That is a Mr. Hamming, it's a Mr. Has thought of that first time a while ago, and that is there for the Hamming distance. And for binary strings, you just XOR them, and then it's simply the number of different bits into strings, obviously. Now, this is the basic question. You have a set of sequences and a motif, and you would like to find a motif that has some degree of conservation, either specified like this or like that. So you can do it pattern-driven and sample-driven. What is that? This basically is, let's talk about a certain motif width. W, let's say we look at motifs that are 10 long now. How many possible motifs of 10 long are there in a DNA sequence? That's my first question to you. Combinatorics, I know. How much is that? I want to hear 4 in your answer. 4 to the power 10, I think, yes. So not 10 to the power 4, not 4 times 10, 4 to the power 10. There is 2 to the power 20, where are we? How many possibilities? 1 million, roughly. So it increases rapidly. That's the point I tried to make. Yeah. So if you want to find, is there any 10 long motif that hits the set of sequences enough, you need to try every one of them. Yeah? Generate any of those, slide them over the sequence, and see whether the sequence flares up or not. Right? So that is basically what is being said here. Again, the sequences. One of these motifs, M1 till MW, find a motif with the best match to S. Here you see again how you can define it. Let's say you do this, it doesn't matter. But now you need to find an algorithm that really generates each and every possible motif. This is the number of possibilities. You find the distance between a motif and the set of sequences. And if they would occur in all the sequences, or if the number of differences overall would be smaller than a certain threshold number, you return if you found it, if the motif is successful or not. And that is then if you find it, this is the thing you've seen before, you need a marker theory. Where is that? Yeah? This is the running time. What is that running time? Here K, for some reason, I stole this slide of someone. Here the W is going to put in a K. I don't know why, but this is just a W. So how would this work? For any motif of 10 long, you slide it over each of the sequences. How many steps do you take? Is it the exact sequence length? Not really, yes, because you don't need to stick over the sequence. But anyway, it's roughly the sequence length. So here is what you see, K times N, but you do this for each possible. And here's the K again for some reason. So for each possible motif you do this. Yeah? What is nice about this? You're absolutely sure that you cover every possible motif. So you try really every possibility. That has an advantage. But the expense, of course, this is slow. This is a lot of work. It might namely be that could it be that one of those motifs among the million is not occurring at all in these sequences? Could it happen? Yeah, but you put in work, right? You make a motif that's not even there. And you slide it over and you find, oh no, the first sequence says no, second sequence says no. In the end, the computer says no. So that's work that you did too much, right? And so maybe there's another way of doing it. Who has an idea now from this limitation? You're doing just work that you shouldn't have done because the stuff is not in the sequence. Who has an idea? Zoom in on the stuff that is in the sequence, perhaps? Is that an idea? Okay, let's see. Here you go. Example-driven means, we're not going to spend any work when, it has no use. We really look at motifs that are existing in the sequences. So how could you do that? We have a 10 long word. How could you find out which words are in the sequences? Take a window of 10. Slide them over the first sequence. Hey, this motif is in the sequence. Hey, this motif is also in any of the sequences and so on. Take those. There will be another way of doing it, right? So this is for m being any word of 10 long, whatever, but occurring in some sequence, at least once, you could try it, right? Is that more expensive? Less expensive? What do you think? Is that always more expensive? You have a motif of too long. You have a database with 10 billion sequences of 3.3 billion letters each. Do you think this is faster than just trying the 16 motif possibilities? I don't think so. So of course it depends on the size of your motif and the size of your database that you're wise enough to take a decision there. So anyway, if you do this, you slide with any of these window length over the sequences and you find that. Yeah? And so this is another thing. What's the drawback? Maybe because suppose you say I have a motif and it's not the exact motif that I need to see. If 3 letters are different, that's fine enough. What happened before with the exhaustive way? You try all possible motifs. So your scoring builds that in, that it can be different. Here, you really look at motifs that are there in the sequence. Maybe another average motif, consensus motif, that is not in the set of sequences is the better representative of that motif family, right? You may not be able to get it here. If you want that, you need to go to the exhaustive one, okay? So both come with drawbacks and advantages, okay? But this is in most cases when there are many possible motifs, this is much faster. Okay, so but again, you have this nagging feel like this is all nice and having these motifs, and they will be there in the sequences, they will be short. How biological are they? Maybe I'm just fooling myself. You get a great motif conserved in each of the sequences. Biologically, it's not doing anything. So what is important, and that was already brought up, conservation. Yeah? And how could you probe conservation? Certainly when you have non-coding regions that tend to be not so conserved, you hope to have just local bits that are conserved. You align them between orthologous sequences, yeah? Be they DNA or protein. Yeah? And then you try and find these transcription factor binding sites as an example of a functional motif only in conserved regions. So you only believe that it's functional when you see a degree of conservation between orthologous species, the DNA of orthologous species. And then you will find but there are problems. Even if you have some important regulatory regions, they're certainly not all conserved. So the fact that there is no conservation does not necessarily prove that it's not functional, but it makes it less likely. Okay? And then of course, if you are going to compare, with what are you going to compare it? Just another person? Is that good enough if you talk about, you want to find it for humans, or do we say no, I'd like to go to mouse, to something that's a bit more distant, because then conservation is more meaningful. That depends on the problem you have at hand. What you want to choose. So that's the problem you need to think about. Okay, so there is something that is called phylogenetic footprinting. And a footprint, of course, is something that is put in the sand, and here it means something that is conserved between species. The only sentence that is suggested by phylogenetic footprinting is a technique that tries to elucidate bits that are important. Why? Because they're conserved. What is important is conserved, is the motto here, and so that is what it means. Yeah? So Here you see it's the task of finding conserved motifs across different species. And that, of course, relates to common ancestry and homology and all that. Yeah? And then these motifs have resulted in footprints, and that's where the name comes from. Okay, now, here you go. These might be a few genes here, these arrows. This is the bit upstream. This is a mess. The green bits are normally, it's totally unrelated, or maybe related, but not proving any relatedness. Letters are entirely different. But somewhere there are these binding regions we would like to find, and they might have a degree of conservation, the yellow bits here. So what would be the first thing to do? Get a splendid alignment, align this, and that will be fine then. And then you can just look to what extent is it conserved. But the problem, of course, is to get an alignment here with these green bits that are entirely different bits of sequences will be difficult. There was again this program which is more a toolbox than a single program. There is a little tool in there called motif-based alignment that you might try.
Was it in Prague?
That was in Prague.
Yeah, I thought so. I heard it before somewhere.
I don't know. Prague?
Yeah, I think so.
Maybe.
Sounds like a good method.
It's the chocolate from Brussels. Yeah, anyway, so, but if you don't want to go into the trouble of aligning and so on, there is also technology that tries to do this alignment free. That's what we will talk about as well. Let's here take a first method that used alignment. This was, I think, a Nature paper. Here you go. A while ago already. My God, Stone Age. But anyway, you were all already on the planet, right? Promise me that.
Stone Age.
You did. Okay, good. You can talk. Good. So, see, he's older than that birthday here, I think. But anyway, he wrote the paper then. So this is basically what he did. He wanted to find out, to find as many reliable or putatively functional binding regions in DNA sequences, and he probed 4 different species to do that. Of course, he was on about the human sequence. He wanted to learn about that, but to get information, he included the mouse, the rat, the dog DNA sequence. He then extracted genes, there were known genes, and then, so they were known, but how the transcription happens and the regulatory control happens was unclear, so they took 2,000 positions at either side of this gene and said, this is the region where the binding site should fall, so that is what I take as my input, and now I will develop some technology to find conserved, small, short binding sites. And he did it in an exhaustive way, as you can see. So, a few things I need to tell you. So basically, he zoomed in on motifs because normally transcription factor binding motifs are 6 to 18 long, so that's what he took. Of course, he looked in human regions, that was the focus. And he established a sense of motif conservation score, and that's pretty simple. What is a conserved? A conserved occurrence it was a motif that happens exactly matched, aligned in all 4 species. All 4 have the exact motif. Great, that's what we call conserved. Okay, and then he said, okay, it might be that I find a motif, and in this position in the multiple alignment between the 4 species it's conserved, but there is another 2,000 incarnations of that same motif in the human, and there it's not conserved, right? And that's maybe telling if they're all conserved or not. So he established the motif conservation score, and that is just the number of conserved occurrences, how many of these motifs were conserved, divided by those motifs happening in the human DNA that is not totally conserved. Yes? And then all he did was really quite simple. He established, and then he said, okay, this is a score, but when will I trust this score? And then, when you're in trouble, you always ask a statistician, and you know what the statistician always will say, you need a null model. You need a reference model. Okay, there you go. You did it like this. I think you can criticize this or say, is this the way to do it? Personally, I wouldn't have done it like this, but this is how they did it. And they wrote the Nature paper, so they're right. So basically they said, okay, we take 1,000 random motifs across the human DNA. And then we do this 1,000 times, and then we extract, of course, at these loci, so eyes closed, pick 1,000 positions in this alignment of 4 species, extract the human sequences at these loci for the length you're on about, then you scramble them. So you take these and then you scramble them. You say, look, this is really found, but now I take the same sort of complexity, I scramble, I change the order, but not the number of letters in the motif. And then I calculate of those the same score. And then I have it 1,000 times, so I'm able to calculate the mean and the standard deviation of those standard motifs. And this gives me my background information, what to expect. And then you can say, calculate of these scores, the Z scores, given that I would just, what would I have done? I would have said, okay, I'm on about a certain motif, scramble that motif. Why go to all these other motifs? Anyway, this is a weaker reference, a less stringent reference than the one that will result in what I just said. But, you know, they did it like this, they probably wanted some good scores. And how could you get good scores? You have your thing you want, score high, or, you know, suppress your random model, of course. Anyway, maybe that was the thinking, I don't know. Anyway, this is just a way to see clearly where you can see that they use this, this idea of conservation to get a handle on the importance of regulatory, in this case, regulatory binding domains. Okay? What's the time? Am I faster or slower? No, pretty good. Want to go on? I don't think so. No. Okay. Let's call it the end of lecture then.